Hi everyone. I thought it would be a little bit easier if I just walked you through the FRQ answer key. It's a nightmare to try to grade them, so a lot of times if I made comments in your AP Classroom on the FRQ questions, then I just said kind of look at the answer key to compare. I did leave comments in each and every one of your kind of FRQ sections, and I highlighted which question or subpart that you might have lost points on. So as you guys move forward and do test corrections, which I will communicate about later, yes, you can do test corrections. You can use this to kind of help guide you in where you might have gone wrong on some of those answer choices. So I'm going to minimize myself. I kind of wrote out what the correct answers should be and put them here. So for example, some of you might have had the two first questions flip-flopped. One of them was related to the cladograms and one of them was really related to the table that's right in front of you here that you needed to do. So the first part of the one question was asking just in general about what is a measure of fitness. And so you should have had something along the lines of that we measure true evolutionary fitness as a function of reproductive success. So they are surviving and reproducing or leaving more offspring that can then have more offspring and so on. That was worth 1 point. A lot of people might have lost some kind of partial points on this particular graph. Some of it was silly things and some of it was more big picture things. And I know that it was complicated material. This graph also does not really show the standard error of the mean, which are those little plus minus bars that you could have there, which kind of amplify the score potentially even a little bit more. But key features here, a lot of people drew line graphs of various sorts. AP pretty much wanted you to draw a bar graph. So if you drew some kind of line graph instead of a bar graph, then you lost a little bit. One common thing that a lot of people did is they might have switched alleles and populations. So you might have had alleles along the bottom, like allele 1, allele 2, allele 3, and then had 3 populations. But the major overall goal of this whole thing was to compare the different populations. So it makes a little bit more sense to put the 3 different populations across the bottom, and then you can see the distribution of alleles in those populations. And I also know that this is a hard one because it's not just 2 alleles. There's actually 3, and then that throws a whole bunch of craziness into the mix. So you might have lost some if you had switched that. Some of you lost an easy point by the title, or you didn't label your axes in some way. So those are just silly things that AP will always ding you on if you don't do that or add that in. And for me, the title, I just copied it straight from the actual question or the table that had that information. I gave a key so you can kind of see the distribution of those 3 things over time. So just being able to reconstruct that was a total of 3 points. I kind of equated scale and unit to whether or not you had a title and a key of some kind. You should have had your axes labels, and then it wanted a bar graph. In terms of other things too, it also asks you a little bit about, are the populations or are the alleles statistically different? And yes, they are, because a lot of them have drastic differences, and the standard error bars, if you look at the actual numbers, don't overlap. Okay. In terms of which one has the most heterozygotes, according to my graph, it would be C. And the easiest way to talk about that is just because they have the most even distribution of the 3 alleles. The difference between them is not as significant as it is for population 1 and 2. Both in population 1 and 2, there's a significantly higher number of allele 1s compared to the others. So in terms of heterozygotes, in this case, it could be a mix of 1 and 2, or 2 and 3, or 1 and 3. It's just meaning that it contains 2 different alleles. That's the true meaning of what a heterozygote is. And the likelihood of that happening is way higher in population 3. That was worth a point. And then came the calculation part where everyone was annihilated. I think it's because there were 3 alleles. Some of you might have just lost a half a point. because you had the right idea or the right formula. You might have just forgotten to multiply by 2 or something. Some of you did all kinds of crazy math. But, oh, I do want to mention too, some of you said that you couldn't really tell because it doesn't tell you what is dominant and what is recessive. In this particular instance, that doesn't matter because anytime you're figuring out the heterozygote, you multiply P and Q together. So it doesn't matter which one is which in this instance. And it's specifically telling you allele 1 and 3. So essentially, those are your P and Q, whether it's P and Q or Q and P. To figure out the heterozygote, it does not matter. And these are telling you allele frequencies, not genotype frequencies. So because they're allele frequencies, they are correlated with P and Q, not P squared and Q squared. So you literally just had to take 2PQ, 2 times the population of 1 and the population of, or excuse me, the frequency of 1 and the frequency of 3. So in this case, it was 0.6 and 0.25, according to my little graph here, which ended up being 0.3. So that's probably the one that most people got wrong. Scrolling down to part D, most of you got this part correct in the sense of population 2 is probably going to be that most highly effective because if we look at the graph, it has the least allele diversity. Meaning that there's not as much of a distribution of alleles in population 2 compared to the others. So if you are shifting away from something that is kind of advantageous for allele 1, well, that's going to have a drastic effect on population 2, and it will probably go down because it's the least fit, so to speak, to adapt to any kind of change in the environment. 3 would probably be the most successful, but 2 would probably see a decrease in individuals because of the unequal distribution of alleles. So you should have had something in there. If you had the right prediction, that got you a point, and then the explanation of that got you a point. Now, for the other question, you should have said that the LFS2 had the most amount of differences. If you count up the letters, there are 13 letter differences between that and the reference animal. So if you said that, then you got a point. Keep in mind, I didn't mark down if you didn't write a complete sentence, but AP wants you to write in complete sentences. For part B, some of you had some crazy cladograms. You might have had it backwards, meaning you had the dugong here at the top and then you worked your way down. But please keep in mind, time is going from bottom to top. And the out group is kind of the least evolutionarily advanced or contains the least amount of characteristics or is the most different from all the others. So therefore, it's always going to branch off first. And then if you count up the number of differences, Alphas is the next one, and you could kind of have that information from part A is the next and most distant compared to the reference one. These two branches you could have had either way, which is why I have this little note here. So a lot of you put mammoths here and Lexodonta here, or mammoths and then African elephants, and that's okay too. So whether you had it this way or flipped around, you can kind of rotate around this node and have it be equally correct. Some of you are under the misconception too that because mammoths are extinct, that they had to be farther away, but that's not necessarily true. Just because an animal is extinct doesn't necessarily mean that it's far distantly related compared to something else. So don't kind of give in to that misconception. This whole thing was worth 1, so depending on how badly you messed it up, you might have gotten a 0.5 off or 1 off. C was a highly missed question because a lot of people delved into the idea of fossils and how we organize fossils and how we can compare fossils. But the question really was asking you, how can we use molecular data from fossils? So it's not actually asking you to compare the fossils themselves. It's really asking you if you compared molecular data. So that's DNA and RNA and or proteins. So looking at that, you have to really think about the idea of conserved proteins. So you can take DNA from those fossils and then run the DNA and see how closely related it is to other organisms that are alive today and then begin to organize it. So some of you had excellent information. It just wasn't the information that the question was asking for. Same thing in D. It talked about how is it possible that you have so many different morphologies or physical appearances based on something that is closely evolutionary related. And many of you dove into homologous structures, which is totally true. Those are homologous structures if they do have a common ancestor and a similar origin. But the key part is how. How do homologous structures come about? How do they actually become different over time? And so this question is really a general natural selection question. If that original population was kind of separated out into multiple populations, I think the Galapagos finches is a perfect example. Well, then in each one of those environments, there's natural selection taking place. So some of you mentioned mutation in your answer, and that's fine too, because that happens. But the idea is that natural selection is the how. Over time, those different organisms are adapting, and that will then yield multiple morphologies, even though they have a common ancestor that might be recent. So depending on where you went with that, you might have lost a little bit. The last question that was in that FRQ part 2 section was worth 6 total points. You basically had to go through and pick 3 things and then a strength or a weakness. Now, I haven't graded it yet, so I don't know what kind of craziness you have in there, but things that you could have mentioned. Fossils. The good thing about fossils is that they can show you time. You can carbon date them. You can organize them in a relative way. It does give you information about extinct species. One of the weaknesses of it, though, is that we have an incomplete fossil record. So we don't actually have fossils for everything. That makes it kind of hard. Second thing you could talk about is anatomy or morphology. So this is that idea, again, of homologous structures is a strength. Problem is analogous structures. Those are the ones that result from convergent evolution. So those are the ones that end up looking similar, not because of a common ancestor, but because of similar environmental pressures. You could have talked about embryology, the idea that very, very early on in our embryological development, a lot of things look similar. And that is one way to create evolutionary relationships. The problem with that is that way beyond, or at least a little bit beyond that super, super early development, then we do really start to look different. So only looking embryologically, you can't always capture that perfect moment to see how similar we are. Molecular data, as referenced above in the earlier problem, you could use. It's the most accurate way to track differences because you just count up the DNA differences or protein differences between them. The problem is that we don't have that necessarily for extinct species or things going super far back where we can't actually collect DNA. And then the last thing, which we really didn't talk about, but in theory you could have thrown it in there, has to do with behavior. So there are certain instances where behavioral traits are genetic, meaning the organism is born with those things like bird songs, potentially, or frog calls are another thing. The problem is some behaviors are learned. So you can't really parse out genetic similarities in behavior versus learned behaviors once an organism is already alive, which would not suggest an evolutionary relationship. So you could have had any combination thereof of 3 things and a strength or a weakness. In my opinion, that was the softball question that I meant to send your way to hopefully salvage any and all points because I thought it was super easy. Hopefully it was true. Like I said, I haven't graded it yet, but use this answer key. I will post it in Buzz and send this video out so you do have it when you are making test corrections if you so choose. Thank you.